So greetings and good morning to everyone. Welcome to our Quest webinar. Stands for Webinars Empowering Science and Technology. For those of you that aren't aware, this webinar started around the pandemic. So we've been doing these West Hour webinars for 2 years now. So for those that have repeatedly joined us, we thank you, and anyone new, we're excited to have you join us. Today, Dr. Viet Nguyen will be speaking about rethink cell therapy with lipid nanoparticles. We're really excited about the topic. Before we get started, just a quick introduction to the North America West team. So my name's Kim Killian, and I'm the Regional Sales Manager for the Southwest Territory. I'm located in San Diego, California, and I'm joined by Viet Nguyen, who is one of my field application scientists. Viet, who will be actually presenting to you this morning. He's located in the greater LA area. And we're excited to introduce you to our newest field application scientist, Dr. Ingram Vargas, who is also located in San Diego. And then, of course, we have our Northwest team with Angel Galarza, who's the Regional Sales Director located in San Francisco. And then we also have Ian Villamagna, who is Angel's field application scientist, and he's located in Seattle. Ian won't be joining us this morning. He's taking a well-deserved paternity leave. And I think that's probably everything we need. Also, like I said, we've been doing this for quite some time. We have journal clubs, and we also have our webinars, and you can check them out on our YouTube playlist located at that link above. And again, we're under West Hour. So I think that might be everything. Just a reminder, we do have a journal club coming up on the 29th of July, so that's next month. And we always do them on a Friday, and we will be sending you out information about the topic and the journal so you can prepare in advance. But I think that's everything. I'll stop talking now and I'll hand it over to Viet. Thank you.
Thank you, and thank you, Francesca, and thank you everyone for joining us today morning, right before July 4th weekend. Today we'll be talking a little bit about really T-cell therapy with lipid nanoparticles. At Precision Systems, we always like to start with our vision, which is to accelerate the creation of transformative medicine that significantly impacts human well-being, essentially providing solutions to our customers for their nanomedicine development needs in a way independent of the stage they're at. I would say that the space that we are seeing most activity in, and we're providing a lot of solutions in these days, is the genomic medicine space, and that's a really exciting space to be in, just due to the versatility of these types of constructs, the versatility of and the ability to target virtually any gene and design your drugs to specific diseases. And the recent COVID pandemic has also validated that these are manufacturable and clinically validated. However, they're inherently unstable, and they do need a delivery vehicle. And one of the most modern ways to deliver these is using nucleic acid lipid nanoparticles, or LNPs. These are inherently, multi-component systems. They can be tuned. These components, the excipients, can be actually tuned and manipulated to be efficient in delivery in specific systems. And that's really important, because depending on the disease that you're treating, you really want to treat and deliver these drugs in different locations, different spaces, in a different manner. And probably the biggest example of this would be, we know the current COVID mRNA vaccines, for them to function properly, they do need to engage the immune compartment. However, if you're looking towards a gene therapy, a protein delivery, you actually do not want to engage a protein compartment, so you can adjust this composition, the excipients, your carrier vehicle, to lower that engagement with the immune compartment, so that the protein that you produced is not being attacked by our body. Similarly, there's considerations, in the ex vivo space. If you're looking towards cell therapy, the number of cells and the viability of these cells matters a lot. In general, you'd be, extracting T cells, sourcing them from the patient, engineering them, and injecting them back into the patient for the cells to be a drug, right? So the raw number of these cells and really how safe this method is, how safe it is to use, can matter quite a bit, because your raw amount of drug will just go down if you do lose any of these in the process. The current T cell space is also comparatively young. It's the first FDA approved T cell therapy, CAR T cell therapy drug was approved in 2017. And we're seeing a lot of movement in this space. There's definitely a lot of interest. Different programs moving towards clinical trials. Another challenge is also related to that raw number of cells, in that some of these patients that you would want to take these cells from and use them as a drug, they're very sick, and you just have a hard time getting a lot of cells. These cells aren't healthy, so they're hard to expand to get enough of this, your drug molecule, literally your drug, the cells themselves. And therefore, there's also interest in developing allogeneic approaches to source these cells from healthy patients and reduce the immune burden of them and use them as almost an off-the-shelf drug for other patients, which involves more manipulation of these cells. The current state of art, as far as methods that you can use to modify these T cells in the cell therapy space, really span viral vectors and electroporation. These would be the ones that are commonly used nowadays, especially viral vectors is the one that's used in the currently approved drugs. Again, they come with a set of advantages: efficiency, ease of access, ease of use, but also a set of disadvantages such as high manufacturing costs and potential mutagenesis. So there is still a need for other methods to again, there's room for improvement in these. Electroporation, on the other hand, tends to result in lower cell viability, so it's a bit challenging when you're trying to preserve that cell count, the raw amount of drug cells that you have. And which takes us really to, our suggestion really to switch that method of delivery and thinking about utilizing lipid nanoparticles to deliver these drugs in the cell therapy space. Again, LNPs have been clinically validated in other spaces. They do use an endogenous uptake pathway, and we'll highlight some advantages of these LNP workflows in the next several slides. From validation, the first LNP drug that's been used in humans actually was approved in 2018. That was an siRNA drug, an IV drug, followed by the COVID drugs that we are all aware of. And really, it's a space that also is seeing a lot of movement, which is why it can be very beneficial to utilize this emerging technology in this cell therapy space. These particles inherently, at least in their current embodiments, they are taken in cells via endogenous cellular transport pathway. That uptake is greatly enhanced by a LDL receptor-driven pathway, and therefore this this uptake mechanism is pretty well described and well understood and can be tuned. The compositions of these LNPs can be tuned to increase and modulate this uptake, as well as an endosomal escape, so that the payload gets into the cytoplasm of the cell. But this all takes us to our offering, our solution for this specific space. So our GenVoy-ILM T cell kit for mRNA, which I'll spend the next several slides discussing. I also should have mentioned that from the start, but if you do have any questions, please feel free to add them to Q&A. My colleague Ingram will be watching out for them if I can't see them while speaking. Okay. The T cell kit for mRNA is essentially an off-the-shelf lipid mix, with those canonical lipid excipients that is a ready-to-go kit that can be used with any RNA of interest, nucleic acid of interest, to encapsulate using our instrumentation. This is our discovery scale instrument that this kit is designed for, and with the goal of essentially providing easy, simple workflows to be able to start making these LNPs. From a process perspective, or essentially, when designing this kit, we wanted to not interfere with the should I say standard way of that these cells are sourced from humans, expanded, modulated, engineered, and then used as a drug. So this method is in a way tangential to that process, where you would, in tangent with the cellular process, you would essentially make these particles, and the workflows here take minutes with some time for characterization. But this can be seamlessly integrated into workflows involving typical T cell culture expansion. From an activity perspective, when we're looking to use these constructs to deliver mRNA, in this case, the examples here are two different types of RNA, EGFP constructs, so just a fluorescent protein, and slightly more interesting functional construct, the CD19 CAR mRNA. And you can see that in both cases, the transfection efficiency in our hands with these experiments has been much higher using nucleic acid LNPs compared to a more traditional method, electroporation here in our hands. You see that expression, is still percent transfection is still high after 48 hours with both of the constructs. From the viability perspective, this is the point where it's very important, as mentioned before, for your method of manipulating cells to not significantly affect the cell health, because the raw amount of cells is already low, and so any losses here would result in losses of the amount of drug you have, the final drug, which are your cells. What we see here is essentially high viability of cells following the treatment with these nucleic acid LNPs. Still the same two kinds, one with an EGFP mRNA, one with a CD19 CAR mRNA. We saw expression on the last slide. What we see here is that this treatment is pretty benign to the cells and results in a much lower loss in viability of these cell cultures compared to electroporation in this case. From a drug development perspective, though, we like to have our drugs to have a dose dependence, so you can actually fine tune how much expression you get and how much you dose to get that expression. So we see that here. We see that here in terms of a dose escalation study, ex vivo. This is all in vitro. Looking essentially at the fact that we have a dose-dependent increase in the expression of this case in GFP signal. This expression is good. It's a very sharp peak in these cases, and again, minimal losses to viability at the higher end of the dosing regime. And this holds true also for the CD19 CAR construct. So similarly, we see the dose dependence of the transfection efficiency of the really raw signal you see from the cells, with good expression and minimal hit to viability. All important in this cell therapy space. Interestingly, these cells, when engineered, and when we co-culture them with tumor cells, they're functional. So we can make we can engineer cells to be functional and kill selectively CD19 positive tumor cells in this case, as showcased here, essentially showcasing that against a CD19 positive tumor cell line, we see a very efficient killing, which really doesn't work if the tumor cells don't have the CD19 receptor here. So this is specific to that antigen, to that engineering process that we've employed to modulate these T cells. You might ask, though, this is the discovery kit. You might ask, what's next? What happens if I need enough cells to treat a patient? And we actually have solutions here too that go in tandem with our instrumentation. We do have a line of instrumentation for those who aren't familiar with us, and our preclinical instrument also. We also have this kit available on our preclinical instrument that allows you to make much more of these LNPs and enables you to treat much more cells, showcasing essentially the scalability of this manufacturing process. And it's honestly something that's been already showcased with the COVID vaccines. LNPs are manufacturable. What we're showcasing here is that the kit that we've developed for the T-cell therapy space is also manufacturable and scalable. Showcasing here particle quality, so some hallmarks of good particle quality, good size control, and polydispersity index, so very monodispersed particle populations with good low size, and high entrapment efficiency, high encapsulation efficiency, so the amount of nucleic acid that's inside the LNPs is high, and it's in line between the two instruments, so it's comparable between the two scales that we're making these particles at. Similarly to the data before, the CAR expression. So this is a mRNA delivery protein expression. We see both the discovery and the preclinical instrument, our Spark and Ignite, result in particles that can modulate cells, T cells, primary T cells, to the same degree, both resulting in high cell viability and efficient tumor killing, essentially the same experiment that you saw on the previous slide. We see that high efficiency in particles made on both of our instruments. More interestingly, though, especially pointing towards that allogeneic cell therapy space, is our ability to utilize this kit to deliver gene editing machinery, namely CRISPR-Cas9. This is a co-delivery of Cas9 mRNA and guide RNAs to selectively knock out, in this case, the T cell receptor. And what we showcase here is essentially the ability of these kits. They come with full protocols for you to be able to encapsulate, deliver nucleic acid, but also deliver these gene editing machinery and selectively knock out specific genes, T cell receptor, or two genes in a row. In this case, TCR and CD52. In this case, it's a double knockout. While again, if you do a T cell receptor knockout followed by a delivery of a CAR construct, your cells are still functional, like we've seen before. So what we showcase here is not only the fact that we are able to deliver gene editing machinery with this kit, We also can do that in multiple steps. The efficiency does go down, but it's possible to do multiple steps of manipulations of these cells, which again just expands the playing field and expands your ability as a researcher to design your specific drug, which would be the modified T cells. From a process perspective, the workflow perspective, both kits follow the same workflow processes. We essentially provide our customers with a full guide on how to integrate this, the making and characterization of these nucleic acid LNPs to utilize in both gene delivery and gene editing spaces. So depending on your process and your desired treatment type, we have protocols to provide you to help you make these particles for that specific process.
Viet?
Yes.
There's a question in the chat.
Oh yeah.
Someone is asking, what is the half-life of CAR expression, and how long the CD8 plus T cell effect lasts, and at what time point is the max expression seen?
That's a great question. These are mRNA, so as you're pointing to, the expression, if you're delivering with mRNA, will be transient. We do see that that the level of expression drops over time. Over a week, it'll be below 20%. The peak should actually be pretty early on, within 48 hours, as far as expression of delivered mRNA constructs, I would say. What we see, though, is that even though the surface density does drop over time, as far as the cell killing activity, we do see it last for longer than the surface expression. But it's definitely something that can be, that could be a concern as far as storageability of the drug that are the T cells themselves. One way to avoid that would be to employ, gene editing, knock-in approaches, in which case, you'd see a consistent expression of those constructs. Hopefully that answers your question. Great. Yeah, and essentially what we've showcased today is that our GenVoy T cell kit for mRNA is essentially an optimized kit that's quite versatile in its ability to deliver nucleic acid cargo, and the kit itself, the LNPs that are made with these kits are highly viable. They're efficient in knocking down genes and expressing genes in a transient manner with mRNA in this case. And from a manufacturability perspective, these are scalable, and scalability of these types of constructs has been also validated, in a clinical stage with other LNP types. From a cell workflow perspective, these fit in seamlessly to T cell manipulation workflows, and really it allows you to another tool to develop new cell therapy drugs. And this is looking more towards the future, as a summary of this discussion today, is really the fact that there's definitely a lot of spaces that the T cell therapy is really interested in, and expression of certain constructs, even transient, can be quite beneficial in designing and screening some of these constructs. Of big interest, obviously, is the gene editing space, which we show a glimpse of here with its gene knockouts using our T-cell kit. But again, this is we'd like to introduce it as another tool in your arsenal to be able to develop these types of therapies and modulate your T-cells as you're developing your drugs. Oh, I see a question.
I can read it to you. The question is, what is a typical time period for incubation of LMPs with T cells to achieve high transfection? And then there's another one. Do you want to answer that one first?
Sure. Yeah, we can answer that. To your time of incubation of LMPs with T cells to achieve high transfection. Honestly, we see the expression pretty fast. You saw the data from 24 hours following the incubation. We do usually in our workflows do not wash the cells after dosing them with the LNPs. So they do stay with the LNPs for that 24-hour time frame until we have that readout that you've seen several slides ago. Yeah, I would say that the payload and the type of payload can matter in terms of how fast you see that signal to reach its maximum. Some types of payloads would be slower to express, especially if you have something like a self-amplifying RNA type of construct that would have a much slower onset, but perhaps longer lasting expression. So that could be a benefit of that.
Okay. There was another question, but you already answered.
Okay, great. Thank you.
The next question was just does the incubation depends on the type of payload?
Oh yeah.
Yeah.
Great. And then I think this is the last slide is just to showcase our ability, our solutions from the manufacturing side. These reagents were designed for our manufacturing platform, and they're designed to be used in tandem with our manufacturing solutions outlined here. Our manufacturing platform spans a pretty wide range of spaces and really raw volumes that you can manufacture. But the beauty of these is that they're all off-the-shelf instruments that you can use with our microfluidic technology to be able to make consistent lipid nanoparticles. So it's really crucial that the formation of these nucleic acid LNP constructs, these tools for your research, is consistent and reproducible, and that's what's really enabled on our manufacturing platform. The Spark and Ignite here showcased today. The data was generated on these two instruments, just due to the fact that the raw amount of particles and doses you need in the cell therapy space, at least, are much lower in a way compared to say, a vaccine, where you need much more material. These are more personalized, so you can think about them as tools for potentially distributed manufacturing of these drugs from a point of care perspective at hospitals. That could be a great direction for the future.
There's another question. Someone is asking, how does the NanoAssembler platform compare to making similar particles with a standard ultrasonic bath method?
Yeah, I would say that comparing the two, usually our manufacturing platform would result in a much more dispersed, much more controlled way of making and assembling your drug constructs. This is especially important if you're working with more sensitive drug payloads, such as mRNA, especially self-amplifying RNA. Ultrasonic bath methods would likely result in higher breakage of these self-amplifying RNA strands or longer mRNA strands that are just much more sensitive, and that would again result in lower quality product coming out. it's likely you'll have a higher polydispersity too. But I'd be more concerned about the drug itself, drug molecule. From the LNP perspective, the drug molecule itself, which is the RNA or nucleic acid itself. Thank you for the question. Okay, if there's, thank you for listening. I'd be happy to address further questions.
Thanks, Viet. Great talk as always. If you think of anything after the presentation, don't hesitate to reach out to any of us. We'd be more than happy to follow up with any additional questions or comments. And also, as we mentioned, we did record this and we will put this on our YouTube channel if you want to forward it to any other colleagues or teammates. And again, our journal club will be the 29th of July. It's always the last Friday of the month. And you will receive more information regarding that to register, and if you want to read the journal in advance as well, which we strongly encourage. But again, thanks again for listening, for participating, and we look forward to seeing you next time. For now, have a great, safe, and healthy and happy 4th of July. Take good care. Bye-bye.
Thank you. Take care.